The overall goal of this video is to demonstrate a high-output multiplex assay to determine the onset of apoptosis as measured by Caspase 3.7. First, hypothalamic neurons are challenged with the apoptotic-inducing saturated fatty acid, palmitic acid. Then, the cell viability of the neurons is measured using a fluorescence-based reagent and a microplate reader. Following the onset of apoptosis, the luminogenic substrate DEVD, which is an amino acid sequence cleaved by caspase 3, is added to the cells. The final step of the procedure is to measure luminescence using a microplate reader to determine caspase activity, which increases in cells that are undergoing apoptosis. Ultimately, a ratio of caspase activity per viable cells can be obtained in less than 5 to 6 hours in a 96-well format.
The main advantage of this technique over existing methods like Western blots or ELISAs is that it is a rapid, inexpensive assay that can directly measure Caspase 3.7 activity in multiple well formats.
This method can help answer key questions in the field, such as determining primary modulators of apoptosis.
The utility of this technique is that this multiplexing can be used as an accurate method of assessing cytotoxicity and viability. Although the assay is fairly straightforward, a visual demonstration of this method is critical because it will ensure that the assay will be successfully completed.
Though this method can provide insight into neurodegeneration, it can also be applied to other diseases and in vitro models such as those used in cancer research.
Individuals